So while I'm getting this all set up and ready to go, because apparently I have to, re-download PowerPoint every single time I use this computer. I have opened up your Unit 1 exams. So if you guys have questions about any of the questions that you may have gotten wrong and don't understand why they're wrong, or don't understand, why the right answer is right, anything like that, you guys, if you decide to look through all that, feel free to email me or whatever, and I'll help you guys understand that stuff if you do want to look through your tests. So I'm not going to go over them. Like I said, I think I've said that already, but I'm not going to go over them in class just because it doesn't end up being a very popular choice. So yeah. Anyways, so if you guys do have any questions about that, does anybody have any like general questions about that by any chance? Okay. Any minute you want to work. I hope that you guys were able to go over chapter 6 or at least a little bit. I know a lot of people like to listen to that on like double time or whatever, and that's fine as long as you guys are getting the information out of it. Do what you need to do. But the reason I care that much, number one, chapter 6 is a pretty difficult chapter for people to get. So if you're putting that one off, I would not do that if I were you. Number 2, chapter 8 relies extremely heavily on the concepts of chapter 6. So as I'm going through chapter 8, if you don't know what's going on, you might not even want to sit through this lecture because it's that much reliant on chapter 6. The other part of it, I think you guys mostly had chapter 6 quizzes. You guys had yours worked, right? You had actual questions on your quizzes. It sounds weird maybe, but if you know anybody in my other sections, I accidentally posted the template for the quiz instead. So it had like one question on it. It wasn't even from chapter 6 or anything. And that was my bad. But you guys unfortunately got the real questions. So they kind of got the pass on that one. But yeah, so you guys, at least if you've done chapter 6 quiz, at least a little familiar with the concepts, I hope. So anyways, DNA, the interesting thing about DNA, maybe it's interesting to you, I don't know. Being a double helix and being double stranded or whatever, and let's say that your thing reads A, T, T, G, I don't know. And then opposite of these, we know A binds with T and C binds with G, so this would be T, A, C. And let's go ahead and just draw these, right? And that's our double strand. And this goes on this way, and this goes on that way. If you don't know what I mean by this way and that way, and them going in opposite directions, DNA is anti-parallel. What makes it anti-parallel? Well, this line here isn't a line, right? We all know that. We know that DNA is made up of nucleotides, and those nucleotides consist of a sugar, 5-carbon sugar, and a phosphate group and a nitrogenous base, whatever that is. The A, T, or C, or G represents whatever that nitrogenous base is. Does that make sense? So but up here, this line though, this is like a sugar phosphate backbone repeating. Whatever, you get it. Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. And the base is attached to the sugar. And that's what one nucleotide would be this unit right here, right? A sugar phosphate and a base. So that's all DNA is made up of. And the reason I'm saying anti-parallel, these chemical structures are oriented one way in one direction and the other way on the other strand. So they are not both all oriented the same way. So the carbons that are in the sugars are what they are basing all of this on, those structures. So at this end, we have the number 5 carbon, and at this end, we have the number 3 carbon. So this is going in the 5' to 3' direction. It always goes in the 5' to 3' direction. So if I'm telling you it goes the opposite way down here, this is not 5', right? Because it's going the opposite way. So this is 3', and this is 5'. You don't need to worry about any of the other numbers that are associated in those sugars, thankfully. All you have to know is that DNA is always going to be operating in the 5' to 3' direction, always. That's the correct direction. And we have 2 strands, and they're oriented in opposite ways. This creates a problem whenever we're replicating that DNA, right? Because we're replicating towards one direction. We open up this massive, massive bit of like chromosomal DNA to start making a copy of it. And we open it up and we start moving with our polymerase in one direction from that. That's going to be where it opens up. That's the fork. But the polymerase only operates in a 5' to 3' direction. It will lay down nucleotides in a 5' to 3' direction. So which of these strands, if we separate them out, and we're trying to make copies of each of them, which of them would be the leading strand? It would actually be this one, because we're going to be adding nucleotides basically in the same direction that the one up there is, 5' to 3', matching up to that, right? So that is great. That's the leading strand. It's the leading strand because it's going to be going straight through. We can have our polymerase just pushing up, opening up all this DNA, and pushing through. Then the other side, though, we're going to be opening that up, but we can only be adding nucleotides in the 5' to 3' direction. So do we wait until all of the DNA is open and then go through with the polymerase on that one lonely single strand that got left behind somehow? No. What we do is, as it opens up, as a little fragment of it becomes available as a single strand, we use the polymerase and go make a part of it, the part that's available. It opens up more, we make the next part. It opens up more, and we make the next part. So it's being made in fragments. That is the lagging strand. So you understand now why one is the leading strand and one is the lagging strand, right? The lagging strand is made in fragments. Those fragments are called Okazaki fragments, okay? We then have fragmented strand associated with that. That's a problem. We can do nothing with fragments. It has to be sealed together. We have to have these bonds between the sugars and the phosphates, those phosphodiester bonds. And so we have ligase, an enzyme that comes together and seals all of those fragments together. DNA ligase, an exceedingly important enzyme. It's like the hero in the background that nobody talks about, right? This is that guy. Ligase fixes all of that mess that you just made with these Okazaki fragments and makes it usable DNA. The other thing is, when we're making all this DNA and making a copy of it, whether you're going on the leading strand or the lagging strand, it doesn't matter. You have to be able to build from something. DNA polymerase doesn't just add on to nothing. It doesn't just make a copy out of nowhere. It has to have some place to add those nucleotides onto when we're having those sugar phosphate phosphodiester bonds happening. So what it has to have are primers, and it uses RNA as a primer. So it's basically the same exact sequence, but an RNA version. So remember, that's going to be U instead of the T for RNA. And then we go back and delete that out and then put in DNA bases instead. So that has to happen no matter what. If you are going, 5' to 3', then making your little copy, or if you're going in the lagging strand, it doesn't matter. Anytime that you're adding on any nucleotides to anything, it has to be added to something. It can't just be emptiness. So we've established that DNA is anti-parallel. So it has these directions on it that we've called 5' to 3', whatever. It's based on the sugar phosphate backbone. They have their sugar phosphate backbones running in opposite directions. We also know A's and T's bind with one another, and the G's and C's are going to bind with one another. So now we know that as well. When we're making this copy, we're replicating, which our cells do all the time, right? You fall down and skin your knee and you need new cells at the surface of your skin. You're going to need to replicate your DNA in order to make those new cells, right? So here we have another aspect of DNA. We're splitting these two strands apart, and one's going the leading strand and one's going the lagging strand. Finally, everything's all sealed together, and we have these two new double strands, right? That's what we would make from this. One of them came from an old strand here and then made a new strand from it. One of them came from an old strand here and made a new strand from it, right? That's how that works. That's called semi-conservative. This is a pretty important concept when we get into talking about PCR. That's why I'm giving you guys the basic primer on DNA right now, because you're going to need to understand this when I start talking about PCR, polymerase chain reaction, and how all of this is going to apply to that stuff. We talked about ligase. If you have fragments ever, and you need to put DNA back together again and seal those phosphodiester bonds between the sugars and phosphates, ligase is the enzyme that does that. DNA polymerase, it only works off of primers. It doesn't just add the matching sequence out of nothing, right? So all of these concepts are important when we get into some of the tools that we use for genetic engineering. Now, we also, so A binds with T and G binds with C, great. These bonds here, these dots here, that bond between the G and C or between the T and the A, whatever it is, these are hydrogen bonds. Also exceedingly important. It's not a true bond, right? There's no exchanging of electrons. There's no giving of electrons like we have with covalent or ionic bonds. No electron business going on here. We are associating purely based on the attractions between positives and negatives and slightly positive, slightly whatever happened with our polarity of these molecules in the A's to the T's and the G's to the C's, right? That's holding your DNA together right now. Your two strands, your complementary strands are being held together by hydrogen bonds. That's what this slide is kind of talking to us about. We have our double strand there. If you heat up a hydrogen bond, whether it's in DNA or in a protein, heat can disrupt that and denature that bond pretty easily, right? So that's no surprise. What else can do that? If we had a pH change, right? Acid, having too many hydrogens in there or too few, if we had a base, all of those can disrupt hydrogen bonds. Here we're talking about heat specifically. So the heat and the energy associated with the heat, you add that, it breaks those hydrogen bonds. Crazy. I mean, I feel like that's crazy to think about, that all of your DNA is just held together by hydrogen bonds. There's other proteins and stuff too, but we don't need to talk about that, thank goodness. So if you cool it down though, we split it apart with the heat. If you cool it back down, because we have the A with the T and the G with the C, always, it's going to seal right back up exactly how it was beforehand. Those are useful aspects of DNA when we get into genetic engineering. Okay. Talking about A's and T's and C's and G's and all these sequences of these bases and base pairs and all of whatever else. Restriction endonucleases are enzymes. Anytime you guys, by the way, because I'm talking about DNA ligase first, right? That was, or DNA polymerase, or now restriction endonuclease. All these ACE that I have on here, those, if it ends in ACE, that means it is an enzyme. You guys remember about enzymes? That these are catalysts. They are not used in the reaction, right? They're not going to be used up or even altered necessarily in the reaction, but they're going to help a reaction go, right? That's what enzymes are going to do. All enzymes are protein. All right. Anytime I say ligase, anytime I say endonuclease, any of that, enzymes, all right? All right, so these are enzymes that are made by bacteria, originally made them in nature, and they made them in order to break down foreign DNA that was being introduced into their genome. Whether it was the viruses that were inserting themselves, which we learned about already, right? Or it could be plasmids that they don't want in there for whatever reason. They recognize specific sequences in the DNA and cut the DNA there, right? They recognize a sequence of about 4 to 10 base pairs. So there's, like I said, specific sequences that they recognize. Originally, this was pretty much the only way we had to cut DNA. So when we get to the later thing, that's way like cooler, you'll understand how like different, how special it is compared to these endonucleases. But this is originally the only way we had to cut DNA. You had to have the sequence that matched whatever repertoire of endonucleases that you had available, right? And you don't always have that where you want it to be. So that's a problem. You can, however, it turns out, engineer things to have a cutting site specifically where you wanted it. So taking a gene and putting it, adding a few nucleotides in there using special primers or whatever, and amplifying that with DNA. Now you can cut it with this nuclease. And why is that important? I will show you why. Why would you want to cut these things? So restriction endonucleases recognize sequences at palindromes. So by a palindrome, what I'm talking about here is like if your first strand said C, T, A, G. Yes, that's fine. I think that's what the example is going to be anyways. This one, of course, C binds with G, T binds with A binds with T, and G binds with C, right? So we know 5' to 3', and we know 5' to 3'. Would you look at that? CTAG. It's the same forward on this strand as it is on the opposite strand. That's what we mean by a palindrome here. Okay. The strands read the same things forwards and backwards. Another interesting fact about these, you take that CTAG, you remember how T's bind with A's and G's bind with C's. This T could bind with that A and that G could bind with that C if you folded it in half right in the middle. So that's another way it's kind of like a palindrome as well. So they're all like that. Some of them cut in a special way where they would recognize this sequence and we would cut this apart like this. Some of them cut like that. Some of them, I know there's another color of marker over here. Let me get it. Some of them might cut like this, right down the middle. Okay, so this is the difference between sticky ends and blunt ends, right? With the one that goes dog leg like that, you have this you still have your double strand over here, right? You still have your double strand over here, but now we've got a single strand hanging out off of this end, and a single strand hanging out off of that end, if we were to separate those from each other, right? And so if we cut that and leave those single little bits, the CTAG, separated completely from the GATC at that point, because that's how it cuts, is through all of that. Now you have a sticky end. So something else, some other gene that you have designed that has also been cut by the exact same enzyme, now it has a CTAG on that new gene and can match up with the original down here, shoot, and then DNA ligase seal all back together. We've inserted a gene into a target DNA. That's where the importance is of those sticky ends. I have pictures, don't worry. I have pictures that make more sense. So usually about the sticky ends themselves, about 4 to 5 base pairs. Remember the sequence that it's going to recognize 4 to 10 base pairs, but they're always going to be complementary tails that we can match up using the same exact enzyme. So here we go, CTAG. We've cut the original DNA with the red and blue. We've cut a plasmid in the purple. They have the matching ends, and we can stick that plasmid into the purple. Now, you might be thinking like, okay, well, if you mix all of this into a solution, because it would be in a solution, that's how we do this. We have a DNA from organism 1 and then the purple from organism 2 and putting it together. How do you know for sure that you're not just resealing the purple back together, right? Like, how do you know for sure? There's ways to find out, and don't worry, I will tell you about that as well. I'll tell you all of that. Okay. You can imagine here that if you had random palindromic sequences, like we have with ECO-R1 or HIND3, those guys up there, that your DNA could just randomly have that throughout it, just because that's how its sequence runs when it's coding for a thing or whatever it is. So it's not super, super rare that you would see something like this. But it's not super, super common either. And because my DNA is different than your DNA, where my CTAG is at in my DNA could be in a completely different place than where it might cut your DNA, because you don't have the same, genes. Like I have green eyes, maybe you have blue eyes or brown eyes, and maybe that's where it would cut based on those genes, right? So you can use that to your advantage, which of course we're going to talk about that. Why wouldn't we talk about that? And that can lead us to developing these restriction fragment length polymorphisms, because it's got to have a lot of words in it. Anyways, these RFLPs are little fragments that you get when you cut DNA with these enzymes. And it can be predictable. From like mine would have a predictable pattern, and yours will be a predictable pattern. Every time that you cut mine with that enzyme, I don't know what it is, ECO-R1 or something. Every time you cut that, it will cut in the same place in my DNA, right? Or in the same place in your DNA, but they're probably going to be different patterns between the two of us, like we said. And this is useful. So we can then do a direct comparison of the DNA between two organisms. If that doesn't sound like I'm going into forensics, it should. It should sound like that, right? So we've already talked about DNA ligase and the importance of DNA ligase sealing the bonds between the sugar phosphate groups, the enzyme that allows us to seal back up the different fragments of the DNA. So if I am telling you, we already talked about the fragments from the Okazaki fragments needing to be sealed up. But then this, when you're pairing up these sticky ends, those have to be sealed up too. So we need ligase there as well. You can't just pop them together and you're done. We need an enzyme to help us seal it back up. We also have reverse transcriptase as another useful tool in a lot of this. It allows us to, and we said before, convert RNA to DNA. What it does for HIV is takes its single-stranded RNA genome and turns it into a double-stranded DNA so it can stick that double-stranded DNA into your genome. So that's what reverse transcriptase does. But we can hijack that to use it to our advantage in genetic engineering. And this is important as well because we probably definitely watch the videos and learn about exons and introns. An intron is something that we have in our DNA. I don't know why this doesn't work. Probably unplugged it, right? Yeah. Okay, whatever. I don't care about that. It's the least of my concerns right now. Anyways, introns. Introns do not code for anything. They're just segments of sequence in our genome that don't code for a thing. So what happens is we make our mRNA, try to make it sound simple. DNA becomes mRNA, and then we read that mRNA and make a protein, yeah? Not with eukaryotes. This is only in eukaryotes. Here's up here, I'll let that speak for me. So these introns, what we have to do is cut them out. The whole apparatus that does this is called a spliceosome. And I'm sure you guys know what splicing is, right? Be cutting out and putting DNA back together. That's what splicing is. So we will splice out the intron and then seal together the exons. That's the coding portion that actually contains the information for whatever you're trying to make protein-wise. Our cells have to do this in a lot of our genes. Make our mRNA, cut out the crap we don't need, and seal together the stuff that we do need. And then now we have a working mRNA molecule. Okay. Bacteria do not do this because they don't have stupid DNA sequence like we do. Theirs is just efficient. But that means that if we put our genes directly into the bacteria, just the way that they are, the bacteria aren't going to know what to do with that, right? They can't make the protein because they have this crappy mRNA with introns in it that they would get. And they'd be like, well, great. I don't know what you want me to do with this. Like legit. So what we can do is take that mRNA that we can isolate from people who have like a healthy version of a gene that we want to study, or maybe a mutated version of a gene that we want to study. It doesn't really matter, right? We take the sample of the mRNA that has already been used by your cells to make protein. It's already been spliced. It's already good to go, and use reverse transcriptase to turn it into DNA, back into DNA. So we're not going with your original genetic code. We're going with your mRNAs that were made, already spliced out, already ready to work, and returning that into DNA to pop that into whatever vector we're using to put it into bacteria, if you're using bacteria, right? So no more introns, great. All right, so we've introduced this concept of we can cut a DNA and insert genes into that DNA somehow, whatever you're gonna do with that. But you can tell already that's probably gonna be a useful trick to have. Useful little trick up your sleeve. That means you could put a plasmid and put the human cDNA, right? You take that mRNA that's already spliced, you make a DNA from that with the reverse transcriptase. You put that into a plasmid. Those go into bacteria, right? A little bit of extra DNA has that gene for insulin. Put it into the bacteria, and then the bacteria are cranking out human insulin, right? That's great. That's where your insulin comes from. If you use insulin, if your loved one or family member uses insulin, if your patients use insulin, that's where it comes from. It's from bacteria that we have plugged in human mRNA, turned into DNA, and now they're cranking that out. Great. But I've already told you that in order to get to that point with that plasmid, you have to use enzymes that already recognize specific sequences. You have to know the sequence, and you have to be able to put that, engineer it into the plasmid, as well as your special DNA that you're trying to put into the plasmid, right? This is all a bunch of work that we're talking about. That's why people do this for their PhDs. So this is great. It's a lot of work. You can only use these enzymes. It's the only thing you could ever use to cut DNA until we discovered CRISPR and Cas9. CRISPR, clustered, regularly interspaced, short palindromic repeats. These are bits of DNA that are repeating in bacteria and archaea. Again, just another way for them to get rid of foreign DNA that they don't want in their system, just like the restriction endonucleases. So that's great. That's what it normally works for them. However, we can alter the enzyme associated with this, that's Cas9, and the CRISPR association, which I'm going into the detail of it, but we can alter it to cut at just about any DNA sequence ever. So altering the enzyme, not the DNA this time. Before we had to alter our DNA to match the enzyme. Now we can change this enzyme and cut DNA at any sequence that you could want to. And I'm talking then about cutting your DNA at any place that you want to and putting in any gene that you could ever imagine into your DNA. That's what CRISPR is. So if you've heard about CRISPR, which a lot of people probably have, there's been like 1 million documentaries about it. That's the concept about what it could do potentially. I'm going to tell you this though right now. It is hard to get a body to change its genes whenever you're already existing as an adult or something like that, child, a functioning being, I will say, you know. If we're talking about an embryo, yes, that's a little bit easier because we've got a lot more to work with. We can change the genetic material there and see the changes be carried out throughout the rest of that individual's, you know, lifespan. But doing it in an adult person is a whole other endeavor. So we're dealing with somebody who has, for example, Huntington's disease. Huntington's disease is a very serious, mutation, abnormality. It's hereditary, where women have it, and there's some sort of issue with some protein that they make. I don't know enough about it other than the fact they die around the age of 26 to 27. You're lucky if you make it into your 30s. And they know it because their mom died too. So they know. Imagine that. And then hearing about CRISPR. And now I can give you a functioning version of that gene or set of genes even doing this. But how do you get it to all the cells in that person's body? How do you get to a nervous system as opposed to, you know, putting it in your bone marrow is a whole different thing. Bone marrow, we all know. You take out somebody's bone marrow and you can irradiate the rest of them with the bad bone marrow cells. And you can even put new genes in that bone marrow, which we're going to talk about at the end of this. But reinfuse that back in. But that's bone marrow. And yeah, it might make the protein that you're deficient in, but if you need it in other parts of your body and those parts of your body making it, how do you get it to do that? That's the problem. That's why you see a lot of these people who like literally garage scientists, literally people you can order CRISPR. You can do it right now if you wanted to. CRISPR, Cas9, you can order a thermocycler. You can design your own primers for PCR and make this work, make this happen right now. Insert genes into your genome using CRISPR in your garage. The problem is it doesn't get spread throughout your cells. And then those cells that you maybe put it into die. Like I see a lot. There was this guy who was trying to save his daughter. And so he was trying to see if he could make it work, giving himself injections. And he's like, yeah, I feel so much better. Yeah, I feel so much stronger. He was, I don't say delusional, but you guys know what I mean. Yeah. But yeah, he didn't have that and it didn't stay. And it was just in like that one area in his leg that he injected it into. So how do you get that to work? Great concept, CRISPR actually helps us in a lot of different ways as far as talking about genetic engineering and human genetic engineering as well, but just not in the way that these people want it to. You can't really do it easy. It's not that easy. Okay, polymerase chain reaction. I keep talking about PCR. You guys have probably had a PCR test done at some point in your life. If you didn't get the PCR test done for COVID, I absolutely guarantee you had one done for something else at some point in your life. If you ever went to a doctor and you were made to wait about an hour, you were getting a PCR test done. That's what was happening. It can take longer than that. It doesn't have to only take an hour. But here I'm telling you that we can go from one single DNA bit, one cell's worth of DNA. You leave one cell up here, somebody gets mad about their test score. They come up here and they kill the professor when nobody's looking. And they left one cell up here as evidence. This can take from one cell and turn that one copy of genome into billions of copies in like an hour or two. No problem. That's what PCR does. Why do you need a whole bunch of copies of stuff? Well, you can't really sequence things or work with things or change them or alter them or put pieces of DNA into new plasmids or whatever else with one copy. You need a ton of it in order to do that. In fact, if you are sick and I were just to say, hey, there's this person's DNA, let's just sequence the swab out of their nose. It doesn't work like that. You have to make copies of it first. The greatest thing about PCR, we're going to replicate, yes, we're going to replicate DNA, and it only takes like a couple hours to get your result, but you can read it in real time. You can get your results in real time. So what they do is they have their A's and their T's and their C's and their G's. Each one has a special fluorescent color associated with it. And whenever the polymerase adds an A, it flashes blue. A T, it flashes green. If it adds a G, it flashes red. Or in a C, it flashes yellow. I don't know. And the machine can read that. So if it's adding a whole bunch of C's, yellow, 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 it can document that as it goes. And literally like sequencing, essentially, proving that as we're adding on these things, it has to be matching up to the gene that you're interested in. It has to be matching up to the gene that you're interested in, because remember, we have to build off of primers. We don't build off of nothing. So we have to have primers in there. We're building off of our primers, and those primers are matched for E. coli or COVID, something like that. Let's say COVID. We have primers in there. They're only for COVID. If you have the flu, you won't get blue, yellow, yellow, yellow. You won't get that. You get nothing, because those primers are so specific for COVID that even if you had flu a million times in there, you're not going to pop positive on the COVID test. That's how specific those primers will be. So it's 30 base pairs long, but still specific enough in that sequence to target that gene that marks for COVID. So specific primers for specific gene. Yeah, so anyways, the machine can basically read that you're giving off all these fluorescent colors going on, and whether or not you will care if it's A or T or C or G, it will at least let you know that, hey, we're adding on nucleotides actively. That means this is a positive. And you will know that before the machine's even freaking like 10 minutes in, guaranteed. That's how useful PCR is. Now they have to let it run all the way through. I hate to break it to you because of protocol reasons, right? But that's the idea. You could potentially do that. We did a lot of this testing when I did food testing. We had in the morning, early in the morning, 6 a.m., we get shipment from McDonald's from beef patties. And we would, break off pieces and grow them up in this broth and then put them in an incubator. And at 8 p.m. at night, like 12 hours later, approximately, I would get them out of the incubator and then start processing the broth where the bacteria would have grown, and then running PCR on this. And in a couple of hours, I could tell you if any of those hundreds of patties that McDonald's sent us were positive for E. coli, right? Almost never, by the way. McDonald's is like the cleanest. I'm just going to tell you that. Applebee's not as much. Just saying. So yeah, we have this amazing tool that allows us to make copies of all this stuff, whether it's the cell from somebody that killed the professor, at our evidence that was up here at the front of the desk, or just that swab of your nasal cavity checking for the DNA for COVID or whatever. So useful. So how does PCR work? It works pretty much the same as the replication, except easier. So bonus, right? We're going to open up our double helix, just like we would in replication. We're going to expose our single strands now as templates to work from, just like in replication, right? We're going to add primers, because we always have to build off of primers when we're doing DNA polymerase. There's an issue though, because the way that we're going to separate our strands to open up our helix, that's not happening naturally like it would in our cells, right? That's going to be because that would be happening in our cells, not in the machine. So how do we force that to happen? We use heat, like we just talked about in the very, very beginning slide, right? So we're going to use heat to break up those hydrogen bonds and open up the double helix. Now, we just said heat denatures hydrogen bonds, and most proteins, their structure depends on hydrogen bonds, right? Their enzyme can't work if it's not folded properly. So now we have a problem. We've got DNA polymerase, ACE enzyme, right? And it's a protein. And so we're going to need to be able to work with it in these hot, cold, hot, cold, hot, cold over and over and over again, but not break down that enzyme. So that's where we get into this concept of Taq polymerase. And Taq polymerase down here at the bottom, they also have another one that they call bent polymerase, but the one I always work with out of old school, I guess, I don't know, but Taq polymerase comes from Thermus aquaticus. T for thermus, AQ for aquaticus, Taq. And these are bacteria that grow in like the deep, deep sea vents where it's like above boiling water. They grow there. These are thermophilic bacteria, so they like high temperatures. And their DNA polymerase in their cells just naturally has to be able to survive that high heat, right? That's just how it has to be. So we can steal their enzymes from them and utilize them in our PCR. It's the same process. It's just now we have one that's heat stable. Also, those primers, remember, we can't yet build off of something. So this 15 to 30 bases for our primer to allow the DNA polymerase to build off of, that can help decide your specificity of whatever you're targeting. And I will have to break all of your little hearts and tell you, if you go to a doctor and you are sick, let's just say general sick, doctor doesn't know what's wrong with you type of a thing, and they swab your little nose and they're going to run PCR on it. That is not a thing by itself. They have to be testing for something. So when you guys go and order your tests for the lab for the patients that you'll be working with, you will be ordering for the flu, test for the flu, test for COVID, but you don't just say, test this, I don't know what it is. Tell me what it is with PCR. It doesn't work that way because you have to have primers that are specific to a thing. And that is the only way that it works. There isn't a PCR cycle out there that can run several things at once. Believe me, they tried. So you have to only test one thing at a time. Otherwise, you're risking a lot of false positives and stuff like that. And how do you know that it's this versus this and that? So you have to do one thing at a time. So if you're like, hey, I don't know what my patient has, some sort of respiratory thing. Let's test for mycoplasma. Let's test for mycobacterium, right? Tuberculosis. Let's test for COVID. Let's test for flu. And whatever else you want to test for, adenovirus. I don't know. You'll have to run all of those tests as separate tests. Or if they have a whole bunch of thermocyclers at the lab, then you're good to go because each one will be doing a different test. But if they only have like one and you're running at a rural clinic and you're lucky to even have a PCR machine or primers for something special, right? They might not have primers for tuberculosis if you're in like a rural clinic. So what do? How do you test for that? You do other things to test for it. You probably aren't going to be testing it by PCR. You have to rely on the x-rays, the chest x-rays. You're gonna have to rely on your acid fast staining, stuff like that. There's plenty of other tests that we're going to be learning in the lab, especially later on in the semester, that hospitals can use in place of PCR, where you don't have primers, or it might actually just be more efficient. Like acid fast, saying that's super efficient. I mean, it's pretty easy to just do that rather than wait 1 or 2 hours for PCR. Just stain it, right? 5 minutes with that carbofusion, and that's it. But yeah, so you have to have your primers. So I do want to make you guys aware of that. And if you are working in a rural clinic, let's say someone comes in and they've got Ebola. I don't know where they got it from, but that you probably aren't going to have the primers to test for Ebola. Now, I doubt Integris even has the primers for testing for Ebola. When they were having outbreaks in Texas and all of that, people had Ebola because they traveled and they came into contact with people as healthcare workers. Typically is how it happened. Then they may have had primers on hand for just in case time. So they usually don't keep that stuff on file. So if it comes in with the plague, you probably don't have primers for the plague at Integris Baptist. Just saying. It's just not common, right? So what do you do in those cases? You take a sample from your patient typically and send it off to the CDC and or Los Alamos, depending on who's doing the testing. If it's typically Ebola or the plague, you're probably sending to Los Alamos, because those could be potential, bioterrorist weapons. Yeah, Los Alamos is interested in that. I don't know if you guys know about Los Alamos, but it's shady. It's shady stuff. You should look into it if you don't know. Oh, it's where they do that's where they had supposedly all of the actual Roswell experiments and stuff. If you read into that, I don't believe any of it. I just want to be clear, but they're shady like that. That's what I'm trying to say. Real shady. Okay, so PCR. We're gonna heat up our strands to separate them. Pretty easy. Where is my thing? There it is. Separate our strands. There they are, separated, nice and happy. Then we're going to cool it down just a little bit so that the primers stick on in their appropriate locations. How do you keep the main strands from sticking back together? Well, you can't, but you're going to get enough copies that it's going to be fine. So that's how this works. You have to have an excess of primers, really, is how this works. So you have enough primers in there. The primers want to stick instead of the strands sticking back together. And then you're going to have your little extension happening. So your denaturation of the strands, your priming, and your extension. Those are your main steps. What comes after the extension? It's going to be denaturation again. That's it. Those are the steps. So denature, prime, make the copy. Heat it back up, spread them apart. Prime, make the copy. Over and over and over again. So one cycle in PCR would be taking that one sample, open it up, make that one copy with the primer, right? That's one cycle. After just 36 cycles, you can have, what does it say down here? Billions, I know it's billions, 68 billion molecules, because of how exponential growth works. That is crazy, just 36 cycles. So if it takes you a minute to go through one cycle, just 36 minutes and you're done. That is nuts. And one of the most useful tools that we have in science and in medicine. All right, so let's get into recombinant DNA technology. We're going to put that PCR to work. We're going to talk about some plasmids and put them to work too. Anytime that you are using recombinant DNA technology, you are putting a gene from one organism into another organism, such as human insulin into our bacteria. That is a clone. That's a genetic clone. And we're regrowing them up. They have that special gene in there. They're all exact copies of one another. There are literal clones as well. So anytime you are cloning, and you're trying to accomplish this, you need a vector and a cloning host. The vector usually is going to be, like I keep saying, plasmids, the most common one, or viruses. We've already said about HIV and how it will insert its DNA into your genome. Imagine if you could utilize that. Replace its genome with a working gene for insulin or something like that, right? They do that. That's real. That's real life science, okay? So you can make the virus work for you. You can also have a bacteriophage and put genes into bacteria, so the bacteria have it incorporated in their actual genome, as opposed to, it just depends on what works best for whatever you're looking at, right? And then that organism will make whatever product that you're targeting. You can use yeast as well. It doesn't always have to be bacteria. So remember, plasmids are those small circular double-stranded DNA molecules. They are not required for the survival of bacteria, but they often carry genes that help the bacteria to survive. A lot of times they're going to have, like it says down here, resistance to antibiotics. And that's useful too. Because if I'm telling you I have a plasmid that I'm working with and I put my gene in that normally doesn't exist in my vector, my nice, happy, clean E. coli, right? My competent cells. It doesn't have any plasmids in it. And we know that because it's been defined. We've already investigated it. Now we have this plasmid and it has antibiotic resistance against ampicillin. So if an organism had that plasmid in it, it could survive in the presence of ampicillin. We can put our gene in that plasmid through our little cut and paste thing that we were talking about already, and then put that plasmid into our competent E. coli cells and plate all of that mess on some plates. If it has ampicillin in the growth medium, only the cells that have the plasmid will grow. And that's how you know which ones have the plasmid, because some of them won't take it up. We know that. So that's how you would tell. We can also use bacteriophages. We already said they can transfer genes to the bacteria that way. So this is an example of a plasmid that does in fact have ampicillin resistance on it. That's in the brown on the left. And then we have this area, this ORI thing, that's just origin of replication. So this is where it would copy itself when the bacteria are going to multiply. But this, LAC-Z alpha. Typically what that is, it's going to be triggered to do something if lactose is there. And if you put your gene in there, it no longer can do that thing. So it breaks up the gene when you insert yours. So that's another way you can tell that you inserted your gene properly. And then also with the resistance, tell that your plasmid got in the right cells. Anyways, so this area has something called a multiple cloning site. These are all restriction enzymes. We recognize them from earlier, I'm sure. With those palindromic sequences, they can cut in those sticky ends. And based on how you designed your DNA gene that you want to put in this plasmid, it might be useful to use HIND3 instead of EcoR1 or something. So that's why they have all of this designed and made up for you. You can order these online. Easy. All over the place. PUC19 is one of the most commonly used plasmids ever in the history of science. So as long as you can cut your thing with EcoR1 at the top, and then you cut your plasmid with that, because it says it can, because they designed it that way, you can put your gene in there. And if you did, it'll break up that LAC-Z alpha. And if you plate it, and we know that it has the correct thing in there because it's resistant to the ampicillin. So that's how all of that works. That's the concept behind it, right? But first, you've got to make your stuff enough DNA in your PCR machine. Okay, basically, what do you want in a cloning host? Now we're talking about, when I talk about E. coli, that's probably the most common bacterial host that people will use in science to produce some sort of product. What do you want in general? You want something that has rapid turnover, fast growth rate, grown in large quantities in normal culture methods. You don't want to be spending a lot of money on special stuff. You don't want to be a pathogen. So normal E. coli that they engineered all of this stuff with is BSL1 E. coli. It's not going to be causing disease in people. It doesn't have the genes for it. So it's non-pathogenic. We want to make sure that the genome is well mapped so we have no surprises going on genetically. And anyways, it has to work well with your vectors, plasmid or whatever it is, and create a whole lot of whatever your product is. Here's an example. I already told you guys about whatever it is, insulin. Here's another example. There's a drug, alpha 2A interferon. They sell it as Roferon A. This is interferon. We've already talked about interferon, and it's that signal molecule that you would send to your neighbor cell saying that a virus is infecting you, right? Anytime there's anything going wrong with DNA in general, or some sort of genome thing, weird stuff going on, you can use interferon to alert your neighbors. And that is what this is for here. So we have hairy cell leukemia and Kaposi sarcoma. Both of these are cancers. Kaposi sarcoma is very closely related with HIV infection. So they need as much help as they can get with their immune system fighting this stuff. So yeah, this is just going to help the body attack the cancer. And there's a lot of stuff you can get that does that same thing. So we made sure we put it in bacteria. We made sure it didn't have any introns and everything. So of course you need, for genetic cloning, you need your original genetic donor gene, of course, and you need some sort of plasmid that you're going to put it in, of course. So we've isolated our gene. We've done that. I'm going to leave you out of the pain of talking about that one. And we've got our plasmid. Here's our plasmid. And here's the little sequence that we're interested in. We're going to cut it there with our little enzyme. I don't know, EcoR1 or whatever it is. And then our gene happens to have that as well. We've engineered it so on its little ends, so we can stick that into the plasmid. Now we have our recombinant plasmid with our gene of interest, and then we'll put it into our cloning host, bacteria, whatever it is. Then it's going to go ahead and, do normal things that it would do with genes in general. Go through transcription and translation, right? Remember transcription? Here it is making RNA from DNA, and translation is translating the nucleotide sequence there into protein. And then that'll build up in the media around it, and you can filter it out and purify it. There's all sorts of ways you can do that too. So that's how we do that. There's something else in genetic engineering called synthetic biology, which is probably about the most terrifying thing you can imagine from any sci-fi movie you've ever heard of. There were scientists in 2010 that made a new bacterial species, completely new. It's not related to anything else that's ever existed, based just on their knowledge of bacterial cells and how they operate. Complete new species of bacteria that can self-replicate from scratch, just using A, T, C, and G. And now they have bacteria that can exist because of that. I don't know what to say about it, right? Like, I don't know how I feel about it, and I'm sure you guys don't either. Building a new organism from scratch, a bacteria is one thing. It just gets questionable, I feel like, really fast. So, cool science, I guess. All right. So you could potentially, though, use this for good. Even if we're not talking about whole organisms, that's the questionable part of it, like introducing a whole new organism to the planet just from your own human mind. But you can potentially make genes this way and design genes that, based on your knowledge of proteins and genetics, could help fight cancer or other pathogens of whatever sort. So that could be useful. All right, that can be done completely synthetically and not building it from, other existing DNA. Genome analysis, talking about ancestry, we're talking about all sorts of stuff, comparing DNAs to one another, including crime scenes, and we're going to get into that. Gel electrophoresis is a tool that they often use for this. If you've ever seen the picture of like the little bars all compared to one another on the chart, that is gel electrophoresis. All right, so how it works. How it works is you have fragments of DNA somehow. Maybe you cut the DNA with one of those restriction enzymes, right? EcoR1, like we've been talking about. So we got fragments. You put them into this gel. It's made of agarose, and agarose is like a sugar that came from agar, but it's like clear. And it's got these little wells in it, and you can inject your solution of your DNA into each of the wells. You've got glycerol to weigh it down and everything. Then there's this buffer that it sits in that helps maintain the charge with the electricity so that your DNA, which we all know, because remember, we've got all the sugar phosphate. This has a negative charge, right? So DNA has a negative charge. So it's going to want to migrate to the positive electrode when this is happening. So as it moves through the gel, the bigger pieces get caught up in the gel and the smaller ones can get through a little bit easier, right? Sort of like if you had if you're playing football and all these big guys up at the front, but if you had like a nose guard that was like this little guy that can just squeeze through everybody or something. So the smaller size ones are going to go faster through the gel, and the bigger size ones are going to be stuck at the top. So when you look at those charts, up at where we entered our DNA in the wells, the ones closest up there are going to be the largest fragments, and the ones at the bottom are going to be the smaller ones. So it's all based on the base pairs. And then you get an image that looks a lot like this. If we were talking about cutting with an enzyme, remember my DNA is different than yours, we might have a different bars as a result. Same bars, but whatever you want to call them. Those guys could let you know, based on your profile, who's who. Don't worry, there's an example. There's other ways you can investigate this sort of stuff. Nucleic acid hybridization and using probes. This is having single-stranded DNA pieces and like heating up your DNA sample and then seeing if you have the gene that is a single strand for X gene that codes for this cancer or something like that. And matching it up. And if it does match and it sticks, it usually is fluorescent or something that you can identify that it was there. So that's nucleic acid hybridization, matching up gene codes to see if you have the gene of interest. We can do this with bacteria to see if they're carrying the gene that we're interested in to make sure that they have been correctly, inserted the right gene into their genome. Do that. So gene probes, basically the same thing, but with the fluorescence. Anytime you have the fluorescence, it's technically a gene probe. They can also be enzyme linked, like what we are going to learn about with ELISA in the lab. That's going to be fun. Anyway, so how we do this, diagnosing things as well as identifying cultures. This can be useful for both of those without having to go through the process of PCR. Just doing this directly. Anyways, this is just showing an image of how that would work with like this filter that you stick on to the cells, in this case bacteria, and then washing it with the probe and then seeing if it's there or not. FISH is a fluorescent in situ hybridization, which just means you can do this in cells themselves. We'll see which cells have the gene of interest. When is this useful? If you have cancer and you want to see which cells have the cancer in it, you can do it this way. It's fluorescent, so the cells that have cancer will be glowing. So that's kind of terrifying, but yes, it works. So that's FISH. Anytime I'm talking about this DNA stuff, I'm talking about, oh, this was 5 base pairs. Oh, this was 10 base pairs. That's easy enough to imagine. But this is 4 base pairs, right? So 1, 2, 3, 4. That's how, in general, we talk about the size of DNA, FYI. So if on the test, I ask you which one is bigger, 10 base pairs, 100 base pairs, 10 kilobases, or 10 megabases. This is 10 base pairs, this is 100 base pairs, this is 10,000 base pairs, this is 10 million base pairs. So the biggest would be this one down here. Now, it's not going to be that easy on the test. It's going to be like different numbers. So don't get tripped up by the numbers as much. If you need to write it out, like say, write out this is 10 and this 100, this is 10,000 or whatever, so when you compare it, it makes sense because this is easy enough with tens, but on the test, people get tripped up when it says like 4, and then the next one says 75 and whatever. So don't get tripped up on that. It's going to be on Unit 2 as well as on the lab exam. People get it wrong a lot, so that's why I'm bringing it up. All right, anyways, it's just showing comparisons between different genomes. Not really that interested in it, but if you are, you're welcome to look it up. All right, so the DNA fingerprinting, using those restriction enzymes, those endonucleases, to cut things up, and then actually proving that this was this person that was here based on their fingerprint and how it ran out on that gel. So we use that concept of the restriction fragment length polymorphism, cutting up DNA and looking at its fingerprint, what mine looks like cut with EcoR1 versus what yours looks like with EcoR1 versus what hers looks like with EcoR1 and whatever. And everybody in here would probably be different. We can look at specific genes, or we can just cut up whole genome. It doesn't really matter. The fingerprint is going to look different no matter what. So here is an example of how this would work. Each of these here where it says marker, these are ladders to give you a sense of the size as it runs through the gels. That's all these are. But let's say you found 2 sources of evidence. Let's say there was, a hair that you left over there, and then, a piece of skin or something over here, right? That's evidence 1, evidence 2, hair and skin. And the professor is dead. So we found those, and it's not the victims, right? So here's the victim. It's not the victim's DNA, so it's probably the perpetrator's DNA. And so you get your suspects and you line them up. Well, I thought it could have been Joe or John. I don't know. And so you look at them, and suspect number 1, well, it looks like Joe is matching up pretty good here, right? Look at that. And these are here. It's on these other ones too. I know it's hard to see, but the computer would be able to tell if you were analyzing it with the computer. But this is a match more so than anybody else is on this clearly. So, hey, now you found your killer. It's Joe. Joe killed the professor. So that's the idea. You can use a lot of this stuff, whether it's restriction enzymes or PCR, all of this stuff can be used to build whole genome maps, basically. So we have found out through mapping our genome and other organisms that we share approximately 80% with mice, 60% with rice, and 30% with this worm, C. elegans, which is one of the most commonly used. Um, it's a worm, it's like a roundworm, but they use it in genetics as like a model. Anyway, so yeah, they'll use high throughput genome sequencing and PCR in order to build those giant massive, we're talking about billions of base pairs and building that up. You can only do so much in a PCR machine. It's not going to build an entire genome there. All right, bioinformatics. This is just using data to classify genes, protein sequences, and what's going to be made from the genome, genomics and bioinformatics, basically what you're going to predict. One of the things that's important that we can learn from genomics is the importance of the single nucleotide polymorphism. This is showing the importance of what happens when one nucleotide is changed. Maybe one nucleotide changed one amino acid that caused the protein to make my hair brown and your hair blonde, whatever. So that could be one example of a single nucleotide polymorphism. There's a lot of stuff like this, I'm sure you can imagine. There are also single nucleotide polymorphisms that are associated with disease. One of the examples that they give us is thrombophilia. It can even be detrimental or deadly to the organism to have one nucleotide changed. Imagine if I had, remember we have codons, I'm sure you guys remember, in our mRNA, our codon might originally be like AGA. It's going to be 3 nucleotides. Maybe that codes for alanine. I don't know. That's an amino acid, right? However, if we change this one nucleotide in the hundreds of nucleotides long gene, and now it becomes UGA instead. So that's our mutation. It's a single point mutation here. This codes for stop. So no matter where that was, and it could be the second amino acid in that protein. If we hit this mutation and it says stop, we stop making that protein. Imagine that. It screws up that whole protein. Just that one nucleotide change. So easy to imagine that could potentially be detrimental to an organism or even deadly to an organism to have one mutation. Some of them are easy, maybe changes your hair color. The other ones could be deadly. Here we have thrombophilia, where we have 1 point mutation. We are changing from the normal GCT, the codon match being, I don't know, CGA, coding for arginine. Now we're changing that to, instead of CGA, CAA, and now we're coding for glutamine. And glutamine is a different amino acid than arginine is. And because of how it interacts with everything around it, it causes you to have excess clotting instead of normal clotting. So if you have excess clotting, you're at risk for stroke, and you're at risk for needing coronary bypasses and all sorts of stuff that could be very dangerous in your life. Yep. Microarray analysis. This is putting a whole bunch of genes. Let's say the gene for this version of breast cancer, and that version of breast cancer, and that version of breast cancer, all throughout on this huge plate. Mix it with a patient sample and see where it binds, and see what version of breast cancer they have, or something similar. Okay, that's what microarray is, checking for all of these different versions all at once. Yeah, identifying subtypes of cancer and treating each cancer specifically based on that subtype. Extremely useful, not wasting your time with all these different kinds of drugs. We already know kind of the use for recombinant DNA technology, including making some of these products that we can use to treat things like cancer. We've talked about interferons. Interleukins and tumor necrosis factor are similar. Okay, they're used by your immune system to fight cancer in general. You can use that. Remicade and Humira are biologics. We have used recombinant DNA technology to make these biologics. They are antibodies that block the function of certain immune molecules so that you are not having an inflammatory response. So it stifles the inflammatory response. Yeah, thanks. And then here are some other examples, erythropoietin and human growth hormone. for treating anemia or dwarfism, respectfully. Then we have genetically modified organisms. This is the part that I have to get to because if I don't talk about this, I'm going to lose my mind probably. Okay. Anytime genetically modified organisms, also known as transgenic organisms, same thing. Okay. They are anytime you have an organism that you change its genetic code somehow, that is a GMO. All right. So maybe it seems simple enough to you. Of course, we know about this existing in microbes, plants, animals, all sorts of stuff. But I mean, have you guys ever, anybody in here ever had the impossible meat? Yeah, or at least heard of it. Yeah, that's a GMO. I mean, that doesn't happen naturally. Yeah, that's a GMO. Go back to the Humira GMO. That came from a GMO. Any of that cancer treatment stuff, GMO. It came from GMOs. Okay. Let me tell you about this. I know, and I'm not judging you, and you don't have to raise your hand or say a word. Okay. I know some of you probably believe you can eat GMO chicken or plants or whatever, and you think you are at risk of getting the genes, the GMO genes that were introduced into the chicken or the plants. That doesn't happen with any other DNA ever that you eat. Why would it happen with this other DNA? It's just DNA, guys. It's literally just DNA. It's not different or separate. It's DNA. It was in other organisms too before it got in this organism. It's not made up DNA. Okay. You can't, I'm going to tell you this, cannot get the genes from the GMO organism. That's not the concern. Is there a concern associated with GMOs? Absolutely, but it's not that. Okay. The concern with GMOs, things like this. We can use these to our advantage, right? If I'm telling you I'm making plants more resistant to insects, what happens if that gets to other plants? Now, you're just like, well, you just said eat blah, blah, blah. Yeah, if you eat, chicken that was GMO'd, that's not going to get to you. However, if a plant gets a virus and spreads that virus to another plant, it could carry that gene over. There is actually evidence of stuff like that happening. Okay? That's a real thing. They did not, accommodate for that. That's a serious problem. Now we've got plants that are resistant to insects that they shouldn't be resistant to the insects, and maybe that seems like, hey, what's the big deal though? The insects could die because they don't have their food if that gets transferred to the normal ecosystem. And then if they don't have their food and the insects die, then the things that eat the insects are going to die. And then what happens whenever you don't have any food on your table because you didn't want to do GMO? Also, there's the thing too. Not wanting GMO means a lot of food not on your table, a lot of products that you're going to use. It means a lot of farmers who saved their kids, dinner that night because they went GMO. It's by saving their crops this way. We're facing a crisis right now in the world, of course, global warming. It's a very serious thing, but we're actually facing the problems associated with that now. Maybe not here in America, but in other countries, they can't grow crops. They can if they have GMO. What would you do? Face starvation, right? So this is what you're literally dealing with this. This is a real life problem. It's a dilemma. Are there, and I'm sure you can tell, it's a fence that you can be on. Like, of course there's plus, and of course there's minus. Just be aware of which one is which and be educated about it. That's all I'm saying, right? Cool. Because it is scary and there's a lot of unknown there. I think that's the biggest problem with GMOs, the unknown. We don't know what's going to happen long term. When this is actually real, there are actually the ability to spread the genes between different plants. Like that's been observed in nature. Like that should be terrifying. How is that going to impact us with our food and our society? Anyways, moving on. Gene therapy for people. This is looking at somebody who did have something like Huntington's disease or something like that. We're looking mostly at deadly mutations here. Not something like you have a little bit of, I don't know, your limb didn't grow quite right or something like that. Because we're talking about genetically changing your body as it is right now. And whatever could happen as a result of that. Again, a lot of unknowns, right? But if you're dying, these are risks that you might take. So we can actually take adenovirus, which is a cold virus, hollow it out, take out its genome, and put in our gene of interest. Whether it's, making insulin when you couldn't do that or something else, I don't know what. Then you would be able to, make that protein for yourself after you've been infected with that virus. Sounds nice. Getting it to stick and maintain that like genetic, new thing. I mean, normally you get infected with adenovirus and then you get cleared out and move on. But how do you get to stick in somebody's genome? What they do is, just like I had told you earlier, they're going to take your bone marrow out. They're going to infect it with this virus with the new gene in it. And then they're going to transfuse it back into you, your own bone marrow. And so there are cells there that are stem cells that can continue to multiply and produce the proteins there and be effective for you there. They don't need to be all over your body necessarily. Are you going to spread that necessarily if you have kids later on? You have this genetic abnormality that could be hereditary. Is that going to get passed on to your kids? No, it's not, unfortunately. It's not getting to that part of your body, not to those cells. It's just not happening. However, you could potentially have genetic gene therapy to an embryo or a sperm or an egg, something like that. Now we're kind of leaning into the concept of eugenics. If you guys don't know what eugenics is, then not just talking about curing diseases, you're doing IVF. You find out that your baby has this deadly disease and you can change its genetics. That's a great thing. What happens if you want to change their hair color, their eye color, their height, their strength? You want them to be an athlete? Easy. What happens then? Is that morally questionable? I don't know. I say probably, but I don't know, right? I don't want to touch it. So that's one of those things that that gets into eugenics. If you guys haven't seen Gattaca, that's what that movie is about. Like Ethan Hawke is like a natural born human, and then his brother was genetically designed, and he wants to go to space, and so he fakes and uses, Jude Law's genetics or something to go to space. And he has a heart defect. He shouldn't have been in that space. Just saying. There's a reason why you aren't going to space, son. I'm just saying. But it's a good movie if you haven't seen it. But yeah, so here's the end of this. We just made it. This will be the thing that you take the picture of. Sorry, because I had to have a little bit of a review of the DNA in order for this to make sense. I hope that was helpful for you guys. I'll post the Kahoot because I know this is a rough chapter, a rough couple of chapters. The next few chapters leading into our metabolism chapter, which is the last one, I think. Maybe not the last one. Yeah, chapter 10 is the metabolism. Anyways, 9 and 25 are easy. I mean, they're pretty easy. They're like nutrition. They're like nutrition. I'll say relatively. Not this is what I'm trying to say.